Compare the pair. Same genes, different fates. Life's rough for worker bees. Imagine you're born into a life of labour. You spend your days cleaning up after your thousands of siblings and defending your home from ferocious invaders. In a few weeks, you'll have worked yourself to death. Your sister, on the other hand, spends her days mating with males who are literally willing to die for the honour. She'll live 20 times longer than you and be proud mother to thousands of offspring, while you died a spinster. See what I mean? It's rough. What led to this cruel twist of fate, you ask? The only difference. Your sister was fed royal jelly, and you were not. Royal jelly changes the epigenetic marks that are added to the queen bee's DNA. These marks are like signposts that determine which genes are on or off. The epigenetic marks on the queen's DNA lead to queen genes being turned on, while worker genes are turned off. And the opposite happens in worker bees. But what does this have to do with us? Well, these epigenetic marks are found in animals as different as worms, mice, and humans. So the same mechanisms occur in our cells, but minus the jelly. All our cells have the same DNA, but by adding different epigenetic marks to different genes, we're able to get cells with different fates, like an eye cell versus a skin cell. I'm studying the epigenetic marks in eggs and how they shape the fate of an individual. I've used a mouse model where I removed an epigenetic mark called K27 just from the eggs, and I found this leads to hundreds of genes involved in brain development and growth being incorrectly turned on in the egg, when normally they'd be turned off. It's interesting because the offspring that come from these eggs grow differently and have altered behaviour compared with offspring from normal eggs. These growth and behavioural traits in mice resemble a human disease called Weaver syndrome. These patients have skeletal abnormalities and intellectual disability, and importantly, Weaver is caused by mutations in the sperm or egg of the parent, and these mutations affect the protein that puts K27 onto DNA. By comparing our mouse data with data from human patients, I'm hoping to gain new understanding on how this disease is arising in humans. Now remember how the epigenetic marks were changed by the royal jelly? Well, similar to that, a range of drugs have been designed to change the epigenetic marks of cancer cells. They're being used to change the fate of cancer cells and save patients. It's an exciting treatment option, but will likely affect the whole body and possibly change epigenetic marks not only in cancer cells. We don't know how they might affect females' eggs or future offspring. So the next part of my work is to test whether a drug that reduces the level of K27 will also result in the same genes being incorrectly turned on in mice eggs. My results will help develop guidelines to make sure patients can access this life-saving treatment, but while also preserving their reproductive health.